people continually joining, at least 30 people listening to you or more. So I'll go on mute and let you get started.
Okay, well, thank you, Dr. Altman. It's a pleasure to be here, sort of, this morning. I haven't done one of these Zoom presentations before, so it is a little bit weird for me, but hopefully it will be at least somewhat informative, and I won't be too vague talking just to my computer screen. Speaking of which, I hope everybody can see it. And I don't know if everybody was as amazed as I was when this picture was published, but I certainly never thought I'd live to see a black hole, an actual image of a black hole. And I'm showing it because while maybe not quite on the same scale, I think the changes that have occurred in our understanding and ability to treat HAE have been remarkable and worthy of revisiting now and again because it keeps changing and it's a moving target. And, you know, as I think back now, I'm not a young puppy anymore, but over the course of my career, I mean, I have seen this disease entirely change in terms of the implications of it and how we approach the diagnosis. So these are my conflicts of interest, and while I'm certainly not unconflicted, I don't think that it will have any impact on my presentation. And what I'd like to do today is briefly cover some of the clinical aspects of HAE, spend some time then talking about the pathophysiology, because that really underlies how we both diagnose and approach the treatment of the disease, and then finally finish up talking about some of the newer developments in the treatment of HAE. I'm going to assume that most people are familiar with sort of the existing or the prior information, so I'm not going to spend a lot of time talking about some of the basics. Although I will start right from the beginning, which is in 1888, when Osler first described what he called, as you can see here, hereditary angio-neurotic edema. And it took about 75 years before we changed the name, but in the early to mid 1960s, people started referring to this as HAE, or hereditary angioedema, when it became clear that it was not neurotic, that there was an underlying biochemical abnormality, and that was the deficiency of C1 inhibitor activity that was discovered in 1963 by Virginia Donaldson. Now, over the past 20 years, the diagnosis of HAE has gotten much more complicated. And what I'd like to do is quickly show you, some of you have seen this slide before, how I break down a classification of recurrent angioedema without hives, so you can appreciate where HAE fits in and what the differential really involves. And so, you know, first we might distinguish, well, is it hereditary or is it acquired? Not as simple as it sounds, but that's a starting point. And then within the hereditary angioedema, the classic form would be HAE with C1 inhibitor deficiency. And we know that there are two types, type 1 and type 2, imaginatively termed. We're all pretty familiar with those. And until 2000, that's what we called hereditary angioedema. But in 2000, a form of hereditary angioedema with normal C1 inhibitor was described. And over time, we've seen multiple subtypes of this that I'll go into in a little bit more detail later. These are either associated with underlying mutations in various genes or at the current time unknown etiology. But what about the non-hereditary forms of recurrent angioedema? Again, we'll quickly break that down to, is it histamine-mediated or maybe more accurately mast cell-mediated, or is it not mast cell or non-histaminergic? And we don't really focus too much in the field on the histamine or mast cell-mediated angioedema. It's usually pretty clear. It's usually pretty easy to treat. We group it in with chronic urticaria and treat it the same way. The non-histaminergic forms of recurrent angioedema, the major ones are shown here. I'm not going to talk about acquired C1 inhibitor deficiency, which clinically resembles HAE C1 inhibitor quite a bit, other than the age of the patient and the underlying problem. ACE inhibitor-associated angioedema is worthy of its own hour, so I'm not going to talk about that. The idiopathic non-histaminergic is interesting and important in that it's not clear, for reasons that I'll mention in a second, that we can differentiate it from HAE with normal C1 inhibitor, unknown type. And the reason for that is because there is relatively low penetrance of the mutations in HAE with normal C1 inhibitor. So unlike HAE with C1 inhibitor deficiency, we can see silent carriers in HAE with normal C1 inhibitor, and that makes it very tough to know, is it hereditary or is it not hereditary angioedema? And so clinically, we often group these, and until we learn more about the underlying mutations, we don't have a good way of putting some of these patients into the HAE with normal category. So the clinical characteristics of HAE are, again, pretty well known, I think, by everybody in this audience. We obviously start with angioedema that's recurrent without urticaria. The angioedema often involves the gut. So when I say angioedema, I also include recurrent abdominal pain. It's a rare disorder. We used to say, and I still feel that about 1 in 50,000 is about the correct incidence prevalence of the disease. The best data we have comes from some small European and Nordic countries, where they now, with nationalized registries and databases, have a prevalence of about 1 in 62, 63,000. I assume they're missing a couple patients. So I think 1 in 50,000 is pretty accurate. Now, the family history, and now I'm talking about HAE due to C1 inhibitor deficiency, is not always positive. And in fact, about 25% of the patients are thought to have de novo mutations. So the disease has to start somewhere. And that means that the lack of a family history doesn't preclude the possibility that this is HAE. The attacks themselves are very variable. They're often severe. They're generally unpredictable, and they're somewhat stereotypical in their progression, in that they increase relatively slowly. And I would say they tend to progress more slowly than histamine-mediated angioedema, but relentlessly over about 24 hours in most cases, and then plateau and slowly resolve over 2 to 4 days. So the total duration of an attack in HAE tends to be considerably longer than mast cell or histamine-mediated angioedema. And that's one of the clues we use when we interview these patients, along with the family history and the location of the attacks. The common ones are shown on this slide. But maybe most importantly, we focus on the response to standard therapy, meaning antihistamines, steroids, and epinephrine. That's very important, both in helping us understand what we're dealing with, but also in terms of the morbidity and even mortality of the disease, because patients really don't respond to any of these medications. Hence, some of them are given these drugs, sent out of ERs, and then they end up dying, and it causes a lot of problems. It's like everything in life, not as clear cut as we'd like it to be. You know, the saying is all bleeding eventually stops. Well, angioedema, plateaus, and it's a little tough, but it's the job of the clinician to figure out, did it make sense that it was a response to the antihistamine or the steroid, or was it just the natural course of the disease? So that's part of the challenge, but it's very useful to think in those terms. So where do these attacks occur? And this is retrospective data, but it has held up pretty well. And the answer seems to be half the attacks involve the extremities, here called skin attacks, and about half the attacks involve the abdomen, and all other sites amount to maybe 3.6% of attacks. So really the vast majority are either extremity or abdominal. And you would think, why do we focus so much on laryngeal attacks? And the answer is shown here. If we look at attacks per patient rather than per episode, what we see is more than 50% of all HAE patients say, yes, I've had at least, I need some of Len's coffee. I've had at least one laryngeal attack in my lifetime. And since every laryngeal attack is potentially fatal, this is why we focus so much on upper airway attacks. They remain a very disturbing and potentially fatal problem in HAE. And I like these pictures. On the top, you see laryngeal angioedema, and on the bottom right, you see intestinal angioedema in a patient with both HAE patients. And it just shows you how similar the swelling looks. And in fact, it's the mucosal angioedema that causes most of the morbidity, but not all the morbidity in HAE. So the laryngeal is obviously potentially fatal and very frightening. The intestinal angioedema can present like a surgical abdomen. Many of our older patients have had unnecessary abdominal surgery, and it's extremely uncomfortable. Women say it's worse than childbirth. So it's something that patients really don't want or ought not to go through. So if I focus for a second on the laryngeal angioedema and the risk of death, this study illustrates an important reason why it's incumbent on us as allergists to make sure that we're diagnosing these patients and affixing that diagnosis to the chart so people know what's going on. So this was a study out of Germany that looked at 728 patients. And what you can appreciate on the left is that the mortality rate in patients who did not carry the diagnosis of HAE, even though they had it, was far, far higher than in those who carried the proper diagnosis. And on the right, Conrad Bork reviewed the records and went over the progress of these fatal laryngeal attacks. And what he found was, just as we would expect, the early phases of swelling were relatively slow. And then, so that's phase 1. And then phase 2, which is the dyspneic phase, when patients start to say, oh, I feel short of breath, that's pretty short. On average, only 10 minutes long. And then phase 3, which is the unconscious phase, very quickly leads to death in these patients. So you have to recognize it. We need to know that they have HAE and not mess around with drugs that aren't going to work, and get them treated before we get into these rapid phases. And as you can appreciate here, the mean age of asphyxiation in these patients who died was not very great. It was, at least from my perspective, it was only 41 years. And in the bottom right, you can see even there was a 6-year-old who died. So young and old can potentially asphyxiate from HAE. So it really is a big deal. Now, let's talk about the genetics of HAE. When I started in the field, there was a lot of controversy. Was it a regulatory problem with the synthesis of HAE, or were there underlying mutations? Well, we quickly found out that it was in fact due to underlying mutations. And as you can appreciate in this rather old cartoon that I made, the mutations are scattered across the entire gene, primarily, as you'd expect, affecting the exons, a number of large insertions or deletions, but also very many point mutations and small indels. I'd particularly like to draw your attention to the magenta area on exon 8. And this area codes for the reactive mobile loop of the C1 inhibitor protein, and I'll come back to that in a minute. And the reason that I put it in a different color is that with only a single exception, all of the type 2 HAE mutations occur in this very small region of the gene. And I'll explain why that is in a minute. So one way to understand what's going on in HAE C1 inhibitor is to study the mutant proteins. And that's what we've done in this slide, where we just transfect cells with either wild type or normal C1 inhibitor, or in this case, a type 2 mutant C1 inhibitor protein, and then look at, in the lysate would be the protein being made, and it's still within the cell, and over time, it's secreted into the supernatant, so you can see it appearing here outside the cell. And what you can appreciate is that the type 2 mutant protein behaves in a very similar fashion to the normal or wild type protein. So why is it a C1 inhibitor deficiency? Well, that's shown in these cartoons. This is a schematic of the C1 inhibitor protein. The reactive mobile loop is shown in red, and it's waving freely above the rest of the molecule. The molecule is very rigid, and it's under a lot of stress. And the feature that holds it in that way is this central A beta sheet, which is shown in blue. And so what happens is a protease that's normally inhibited by C1 inhibitor comes along. It sees this reactive mobile loop as a substrate. So we might call it a pseudo substrate because it's really not the natural substrate of the protease, but it attacks what it thinks is its substrate and actually cleaves a bond between two amino acids. And when it does that, it releases the stress or the tension in the molecule. And what happens is that the reactive mobile loop inserts itself within the A beta sheet and it drags the protease inside the C1 inhibitor molecule, which essentially closes up on it. And so this is called a molecular mousetrap. And it's a suicide type of inhibitory mechanism. So you lose one molecule of C1 inhibitor for every molecule of protease that gets inhibited. But in type 2 HAE, remember we said that the mutations all occur in this region, in the reactive mobile loop. And the effect is the protease no longer recognizes the reactive mobile loop as a pseudosubstrate. So in fact, the C1 inhibitor just floats around in the plasma like this, completely unencumbered. It does not get cleaved, it does not get destroyed, but it also doesn't function. And that's what type 2 HAE is. Type 1 HAE is very different. So here's type 2, which we just saw. Type 1 is in the bottom. It appears within the cell, but never really gets secreted to any measurable extent. And so it appears to be that it gets stuck inside the cell. And we don't know the reason for that entirely. And I'm going to give you a couple different possibilities. But classically, and to a certain extent true, it involves difficulty in folding of the protein. So this is a thermodynamic gradient used to show how small proteins fold. And the free energy follows this funnel. And so the protein, as it's synthesized in the endoplasmic reticulum, essentially naturally falls down this funnel and comes out on the other side as a native protein. But when you talk about large proteins, and in particularly large glycoproteins like C1 inhibitor, and even more importantly, proteins that have a hard time folding because they're under a lot of stress, the thermodynamic gradient becomes lumpy bumpy with these mountain ranges that can trap proteins so that they don't fold properly. And there's a whole set of machinery within the endoplasmic reticulum called the quality control system to try to either bring it up the gradient, refold it, let it try to fold again. Does it get stuck? Does it fold properly or not? And ultimately destroy it if it doesn't fold. And we think that this is what's going on in type 1 HAE. It's simply the mutant protein can't fold properly, and so it's ultimately destroyed within the cell and not secreted. So here, I'm going to go back to the genetics for a minute. This is the classic inheritance pattern of HAE due to C1 inhibitor deficiency, which is autosomal dominant, like all HAE forms are. 50% of the children of an affected parent tend to get the disease, and it sorts independently. So you could have all four, if you have 4 kids, all 4 of them be affected, or all 4 of them be free, males and females equally. It's very simple genetics, very easy to do genetic counseling for these patients. But let me show you a really unusual, interesting case that was published a couple years ago. And this involved 2 sisters in Belgium who were diagnosed with type 1 HAE. By itself, not very surprising. They had a typical history, recurrent angioedema, made worse with exogenous estrogens. Their complement profile was classic. They had low C4, low C1 inhibitor antigen, low C1 inhibitor function. They genotyped these patients, and what you can see is that there was a mutation right here that converted a threonine to an isoleucine. So everything is copaesthetic, there's nothing really to report. And remember, it's 2 different sisters. But the weird part of this is they looked at the parents, because one of the things we stress in HAE is if you find an affected family member, check the rest of the family, because it's so dangerous to not make the diagnosis of HAE. So they checked both parents and a sister, and none of them had angioedema. None of them had complement abnormalities. They sequenced both the mother and the father, and you can see in the same position with this mutation that they had the normal genotype. Now, if it had been one sister, we'd say, okay, that's a de novo mutation, but it's two sisters. So, one possibility, and they're not identical twins, I should point that out. So one possibility was that the father was not really the biologic father, but they checked that, and that turned out not to be correct. They happened to get lucky. In this case, they decided to investigate it, and they got some of the father's sperm. And lo and behold, some of his sperm had the mutant sequence, while the majority of his sperm had the wild type or the normal sequence. So this was an example of mycochimerism affecting the father's sperm. And that's why the two sisters just happened to get sperm that contained the mutation. And so how often we're missing mycochimerism remains unclear. In other primary immunodeficiencies, it's becoming more and more clear that humans are subject to a lot of chimerism. The rate of mutation is constant and surprisingly high. And let's say that it had involved the ovaries of the mother rather than the sperm of the father, they never would have found it. So it's going to be difficult to know how many of these de novo cases are truly de novo in the baby or the child, or represent a chimerism of the parent. But I think it's a very interesting case. Okay, let's talk about another controversy that I grew up with, which was for many, many years, the dogma in HAE was you use the C1 inhibitor level to make the diagnosis, but it has no practical value in terms of understanding the disease. And it turns out that is not true. And what allowed us to see this relationship was the ability to replace a C1 inhibitor up to various steady states and see what the impact on attack rates were. And that's shown in this curve, which has 95% confidence levels in the solid red. The dotted red is the actual curve, and it's the relative risk of an attack. And what you can see is that as the C1 inhibitor level falls, so as you move left on the horizontal axis, the risk of having an angioedema attack smoothly increases. And what this also tells us is, for a long time, we've thought that right around 40%, right down here, of the normal level of C1 inhibitor is pretty protective in patients. And this was really used to validate that prediction. And the higher you can get the C1 inhibitor activity, the lower the risk of an attack. So it is important to know what the C1 inhibitor activity is. And in particular, if you're replacing C1 inhibitors as part of your therapeutic plan, we are looking to see whether this should become a biomarker that should be assessed. That information is not out there yet. I'm not recommending that people do that quite yet, but it's going to be looked at, and it may become important. But it's certainly important in understanding a fundamental question about HAE, which is why do these patients have attacks at all? If you look at normal individuals, you see that, well, the C1 inhibitor level tends to be at about 100% of normal. That's fine. If you look at patients with HAE C1 inhibitor, they're not generally anywhere close to 50% of normal. If they have one normal gene and one mutated gene, you might say they should be at 50%. And if they were at 50%, the risk of swelling would be quite low. But no, the actual level of C1 inhibitor in untreated HAE C1 inhibitor patients, the functional level tends to be about 15 or 20% of the normal level, which gives you a very high relative risk of an attack. And so the question, why isn't it 50%? Why is it 15 to 20% is actually extremely important. And there was an old study done in the mid-1990s in St. Louis out of the Strunk lab that really gave, I think, an important clue about this. And they studied a patient with type 1 HAE whose mutation was the loss of a stop codon so that the mutant protein could be easily separated from the wild type protein based on size. So they were able to look at the mutant versus the wild type protein separately. There was no difference in the amount of message shown in this northern blot, but when they looked at the secretion of the proteins, here's a normal control, very much like what I showed you earlier. Here it is in the cell, and then with time it appears out of the cell. Here's this patient that they were studying, and you see the two proteins. The smaller one is the normal or wild type protein. The larger one with the loss of the stop codon and 54 amino acids longer is the mutant. And so if we look at the mutant protein first, we see that just like the type 1 patient I showed you, it sticks around inside the cell longer, then finally disappears, but doesn't appear outside the cell. But the surprise was the wild type protein, which hardly appears outside the cell, nothing like this. And when they measured the amount, it was like 12% of normal. And so the implication is that the mutant protein is actually interfering with the secretion of the wild type protein. And that's why the level is not 50%, but more like 15%. So this is an observation that I've been playing with for many years and thought nobody else cared about it, so I haven't even published it yet. So I got scooped. A group from Denmark looked at the same thing and published this a year ago in JCI. And what they said is in the normal genotype, you have C1 inhibitor proteins being made in the ER. They basically don't interact and they get secreted. But with a mutation in one of the proteins, that the proteins interact and the mutant protein basically they think forms a complex with the wild type protein, leading to reduced secretion of the wild type protein. We know why the mutant protein might not be secreted. Now, I don't think they're completely right. I don't think there's actually aggregates being formed, but that's a minor argument. And this is the data that they showed suggesting that it was an aggregate. To save time, let's just focus on this example, the wild type plus mutant, where what they saw was the mutant protein, the wild type protein forming these clumps in the endoplasmic reticulum. To me, what they're showing is, yes, the wild type protein gets held up, it's not secreted, we've done a lot of experiments looking to see by various techniques if there are actual polymers, and we can't find them. Here's how we approach this experiment. We do it a little bit differently in that we co-transfect cells. We use a flag-tagged wild-type C1 inhibitor, and we co-transfect with either unmarked, unlabeled wild-type C1 inhibitor or mutant C1 inhibitor. And then the cDNA makes protein within the cell, either purely wild type or wild type and mutant, one of which has the flag tag on it. And then the protein is either secreted fully or not secreted. And the wild type protein, which is what we care about here, is either labeled with the flag or not labeled with the flag, but we can identify the flag label with an anti-flag antibody. So it's a pretty simple type of experimental approach. And what we've found, this is an example here. So this would be just wild type protein, looking at the amount of flag tagged, and you can see there's quite a bit of it. If we co-transfect with an empty plasmid, it makes no difference compared to co-transfecting with a unlabeled cDNA. But if we co-transfect with type 1 mutant protein, here's two different cDNAs, you can see that there's a variable amount of decrease in the flag signal, so that the wild type protein is reduced, the amount that's secreted, and it's not the same for every mutation. It appears to be mutation dependent, but both of them led to a decrease. Part of the surprise is shown here in that we looked at two different type 2 mutant proteins, and the type 2 mutant proteins also decreased the amount of wild type, which makes sense, of course, because they also have very low C1 inhibitor functional levels. So that's a bit of a surprise and does not, you wouldn't expect that if it's a polymer formation. Is there a question? No, okay. We went on, we've looked at monocytes from patients, normals and HAE, and one of the things that we see, we're looking now at the endoplasmic reticulum, and when the ER gets stressed, it gets wider. It basically balloons out. So here's a normal ER. This would be the ER in an unstimulated monocyte from an HAE patient, and you can see how much wider it is than in the normal. And this is a monocyte where we stimulated it with gamma interferon, so it made even more C1 inhibitor, and the ER further basically widened out. And when we measured it in multiple cells and multiple cuts, these are the values which are all statistically significant, showing that what's happening in HAE is we're getting ER stress. And we think that's what the mutant protein is doing. It's interfering with the secretory pattern because of ER stress. And the biggest surprise is shown here. What we found is that if we co-transfect with mutant alpha-1 antitrypsin, that we also decrease the secretion of wild-type C1 inhibitor. But the mutant alpha-1 antitrypsin doesn't have any impact on the secretion of wild-type antitrypsin. And I found a paper in, I think from the 60s or 70s, I don't remember off the top of my head, where they looked at kids with alpha-1 antitrypsin mutations and commented that, interestingly, the C1 inhibitor was a little low in these patients, and they had no idea why. Well, I think this is the reason why, and I think it doesn't cause angioedema because you don't have the complete loss of one of the copies of the serpin G1, but the wild type protein is impaired. Just by itself, it's not enough to cause disease. And so this is an area where we are looking at ways to rescue the secretion of the wild type protein and hope that that ultimately might be a new, perhaps relatively non-pharmacologic way of treating HAE, or certainly a way to avoid all of these super expensive drugs that I'll talk about in a few minutes. The mediator of swelling and angioedema used to be highly controversial. We now know it's bradykinin, and bradykinin in HAE is generated through activation of the contact system, basically summarized in very skeletal form here. Factor 12 gets auto-activates 12A. 12A cleaves pre-kallikrein to kallikrein. Kallikrein does two things. It both activates more 12 to 12A, which further activates more kallikrein, and it also cleaves high molecular weight kininogen, releasing bradykinin. Bradykinin binds to a B2 bradykinin receptor, increasing permeability. Bradykinin is primarily degraded by ACE into an inactive peptide. So if you give an ACE inhibitor, you inhibit this degradation, so you have more bradykinin. We think that's probably the cause of ACE inhibitor-associated angioedema. A controversial issue is whether bradykinin, which can also be degraded by carboxypeptidase to something called desarge bradykinin, which can bind to the B1 receptor, whether this is involved in angioedema. And it remains uncertain. I'll tell you a little bit more about that in just in a second. So the systems, not only do we have two bradykinin receptors, the B2 and the B1, of which we certainly know the importance of the B2 and we don't of the B1, there are two systems generating kinin in the body. Plasma kallikrein cleaves high molecular weight kininogen, releasing bradykinin. Tissue kallikreins, which are very different than plasma kallikreins, they cleave low molecular weight kininogen, which is an alternatively spliced protein from the same gene, and they generate lysyl bradykinin. Lysyl bradykinin acts on the B2 receptor, as does bradykinin. Both of them can be basically degraded by carboxypeptidase to desarge bradykinin or lyse desarge bradykinin, which can potentially act on the B1 receptor. Both are inactivated by ACE, but also by DPP4 and NEP. So what is the potential role of the B1 receptor? And this study appeared just over a decade ago, where they took plasma from HAE patients and put it on endothelial cells and measured the ability of fluorescent beads to basically move across the endothelial cells. And what they found is the plasma taken during an attack in an HAE patient had a lot more permeability than in a resting plasma from an HAE patient. And they did a clever experiment. They took this attack plasma and they added a B2 bradykinin receptor antagonist, and it partially decreased the permeability. They then added two different B1 receptor antagonists, and they also reduced the permeability. And then they combined a B2 plus a B1 antagonist, and it further reduced the permeability almost down to the resting. And so they suggested that, well, they thought that the B1 receptor, through the action of Desarge bradykinin, was responsible. I think there are several problems with this, the most important of which is that Desarge bradykinin is very active on the B1 receptor in rodents, but in humans, it has almost no activity on the human B1 receptor. So I really don't think that their conclusion is correct. However, the B1 receptor signals constitutively. So if you increase the B1 receptor, it will start signaling and will increase permeability. And what we've shown in the past is bradykinin acting through the B2 receptor is able to induce the expression of the B1 receptor, which is not normally present in cells. But this remains to be fully worked out. To the best of my knowledge, nobody's pursuing a B1 antagonist for the treatment of HAE, at least right now. So let me just show you how bradykinin actually increases permeability. Basically, in normal endothelial cells, permeability is maintained through something called the adherens junction, which is primarily mediated by VE cadherin. When you add bradykinin to the system, two things happen. Number one, you phosphorylate VE cadherin through signaling from a bradykinin receptor, and that leads to cleavage and internalization of VE cadherin, which removes this barrier, and it also causes actin polymerization, so contraction of the cells, which widen the gap between the cells so fluid can move through. And very simply, that's how the angioedema occurs, and it's why it's important to treat early in HAE before you get this movement of fluid that can't then easily be reversed. Let me comment on an interesting observation about the relationship between age of onset of swelling and severity. This is data from a survey that we did with the US HAEA, and it shows that most patients start swelling in childhood, about 50% by the age of 10 or 11, but virtually everybody by the age of 20. The median age was 11 years for the first swelling. But importantly, when we looked at the age of onset of swelling based on severity as an adult, what we found was a highly significant relationship. But a Japanese group looked at 171 of their patients, and what they found is an age of first symptom of 24 years as opposed to in Caucasian. This is both European and US data, both suggest right around 11 years. So in Japan, the first swelling occurs much later, and the Japanese clinicians have always said, well, their patients are less severe than what gets described in Caucasian patients. So we've looked at this more, and I'm just showing you data from two SNPs that we've looked at, one in factor 12 and one in high molecular kininogen gene. And what we found is that both of these SNPs seem to affect the age of symptom onset. And this is a statistically significant relationship. And where it gets really interesting is that the frequency of these alleles is very different in East Asians versus Europeans. Look at this difference. So the less severe allele is much more common in East Asians than in people of European extraction. And in fact, when we looked at our patients and just looked at the people who had the less severe allele for factor 12 and kininogen, we got an average age of symptom onset in the early 20s. So I think that it's an interesting observation that may indicate that there's some truth in the Japanese saying that their patients are in general less severely affected. Okay, let's talk about HAE with normal C1 inhibitor. I see that I'm getting behind, so I'm going to speed up a little bit. Right now, we know of 5 different types. There's factor 12, angiopoietin, plasminogen, kininogen, and HAE unknown. The factor 12 was the first described, and in a beautiful study in Journal of Clinical Investigation a few years back, this group showed that the mutant protein was activated too early and high molecular weight kininogen was cleaved very easily compared to normal. So it really said that the mutant protein activated too easily and then cleaved kininogen generating bradykinin. And then a different group from the Netherlands showed that plasmin was able to cleave the factor 12 in this factor 12 mutant protein, but not in normal not in normal factor 12 to any appreciable extent. So maybe plasmin could be the physiologic stimulus in HAE factor 12. And using a knock-in mouse, they were further able to show that the mutant protein, the expression of this directly correlated, in this case, with bluing, which is vascular permeability. So they really closed the loop and showed that the mutant factor 12 directly led to increased permeability, and it's through the generation of bradykinin. A very nice paper, actually. Now, that's the only one we fully understand of the HAE normal, and it's the only one where I think there's really good evidence that it's bradykinin. We suspect that the mediator is bradykinin and the rest. The other genes include plasminogen, which in Germany seems to be fairly prevalent among this group. We are finding plasminogen mutations in the US population, but we don't know what the mechanism is. What I do want to tell you is that these patients have a lot of swelling of the tongue. So if you see tongue angioedema, suspect in a patient who you think might have HAE normal, it might be plasminogen mutation. That's a clue that you should be aware of. The angiopoietin mutation has only been described so far in 2 families in the world, one in Brazil and the original one in Italy. It does follow an autosomal dominant pattern. The interesting thing about this mutation is that it prevents angiopoietin-1 from forming multimers, so it's more monomeric. And the reason that's important is angiopoietin-1 normally binds to its receptor, which is TIE2, as a multimer, and signals in a way that decreases permeability. So if you have the monomer, it doesn't signal very effectively, and you lose this inhibitory signaling and presumably get increased angioedema. And then the most recently described mutation is in kininogen, very close to where bradykinin is cleaved out of kininogen, but we don't know anything about the mechanism yet in this mutation. So the workup of these patients is very simple for type 1 and type 2. Just measuring C4 and C1 inhibitor is sufficient. But for HAE normal, we've got a real problem and we need a biomarker. And that's where we've done some work. This just shows how simple it is to, by measuring C4, C1 inhibitor antigen, and C1 inhibitor function, we can really distinguish HAE types 1 and 2. And by measuring C1q, it helps us figure out who has acquired C1 inhibitor deficiency. But the HAE normal, the current standard is a really poor one that we published almost a decade ago, which is patients with recurrent angioedema, no hives, normal C1 inhibitor, normal C4. They don't respond to high-dose antihistamines, and they either have a factor 12 mutation, in this case you could say any of the known mutations, or a positive family history. It's hard to most clinicians don't have the ability to look for these mutations, and I've already told you the penetrance is low, so the family history is tough in these patients. So we think that we miss a lot of patients or misdiagnose patients, and we really do need a better test. So we tried to make a test that would distinguish it. We looked at a whole group of patients, non-swelling controls, histamine-allergic angioedema, HAE C1 inhibitor, factor 12 mutants, HAE unknown, and the idiopathic non-histaminergic. I won't go into how we phenotype them. It was very rigorous. It was multiple experts, and we had to agree. We kept looking at the case and treating them and studying them until we could agree. And then we combined the lab data with the clinical data. And what we found is if we activated the plasma intensely, we could not distinguish any of the cases as shown in this red line. But we found what we called the threshold dose that really didn't activate the contact system in control subjects or histamine-mediated angioedema. But you can see it did activate the contact system in HAE C1 inhibitor, in HAE normal due to factor 12, and HAE normal unknown, and in the idiopathic non-histaminergics. We showed that it involved factor 12 because we could inhibit it with corn trypsin inhibitor, which is a potent and specific inhibitor of factor 12, and we got no activity in kallikrein deficient plasma. And then when we looked at the predictive value, it was quite good in our sample, 93% positive and 86% negative predictive value. So we're starting another study. This was a retrospective study where we phenotyped them, did the lab test, and then combined it. Now we're going to do it prospectively and see if we can replicate these values. Then I think we'll have a good biomarker. This is just to say that ours is not the only way to approach it. Alan Kaplan published this where he used a biotinylated protease to bind to plasma C1 inhibitor, basically detected it on an avidin-coated plate with an anti-C1 inhibitor. And what they found was that it worked well, and compared to chromogenic C1 inhibitor assays, which is up here, you can see that the separation was greater, and they're interested in looking at this in HAE normal C1 inhibitor. I'm waiting. They haven't published that yet. We'll see if anything comes out of it. Let me finish, now that I'm out of time, talking about treatment. Everybody knows about on-demand versus prophylaxis. I would say for HAE C1 inhibitor, we would distinguish between first line and second line. First line, currently C1 inhibitor, whether plasma-derived or whether IV or subcutaneous, and lanadelumab. Second line would be androgens and antifibrinolytics. First line on demand, ecallantide, ecantibant, or any of the C1 inhibitors. For HAE normal, we don't know as much yet. First line is not great, progestin and tranexamic acid. Second line, androgens and plasma-derived C1 inhibitor. And We use the same on demand for the HAE normal. Again, no good studies. On demand works as shown here. If you treat early, it works better. You know that already. I won't spend time on it. Swelling of the hands and feet cause a lot of morbidity. Patients feel, we feel that it's worth treating. Extremity attacks, it does involve some clinical judgment, but we've published how much disability that this causes in patients, so you should be aware of that. In terms of anabolic androgens, everybody knows Mike Frank's New England Journal paper showing that danazol was very effective, but this was 600 milligrams. When Mike went back and looked at different doses, we no longer use 600 milligrams a day. If he looked at 200 mgs a day, which is the top dose that we use today because of safety reasons, it's not nearly as effective. Androgens cause lots of side effects, whether people discontinued the androgens because they didn't think they worked well enough or they continue on them. The side effects are the same. The difference is the efficacy, which is much worse in those who stopped androgens than those who stayed on them. Here's the average. This is C1 inhibitor given IV, which only reduced the attacks 50% in the trial, but had like close to a 90% in open label use. And when we compare the data, IV C1 inhibitor works better for every level of control than do androgens, but it's still not good enough. And that's what really led to the desire to go to subcutaneous C1 inhibitor, which has been much more effective. In the phase 3 trial, there was a 95% reduction in attacks. In open label extensions, 70 to 80% of patients, after they've been on it for a while, are attack-free over a 6-month period. So much more effective treatment and essentially giving people normal life. Lanadelumab, totally different approach. It's a long-acting, it's a monoclonal, so you can see here it's long-acting. It prevents high molecular weight kininogen cleavage at the approved dose. It also almost completely ablated attacks. And again, we see 70, 80% of patients being attack-free, and the attacks that do occur are much less severe than in placebo-treated patients, and also is really able to give people near normal life. In terms of HAE normal C1 inhibitor, stop estrogens if they're on it. This is the data from progestin therapy, which has been effective, as shown here, in improvement of most of the patients, worsening only in a couple and no modifications in a small number. So in women, you can give a progestin-only pill. This is the data with tranexamic acid. It's not effective in C1 inhibitor deficiency, but in idiopathic non-histaminergic angioedema or HAE normal, it's much more effective than we see in C1 inhibitor deficiency. So we use a lot of it, and I would suggest that it's worth a look. And let me just maybe finish by saying what's coming down the pike. So people may know that there's an orally available kallikrein inhibitor that's in front of the FDA now that appears to work well in a subgroup of HAE patients, not well in another group. And so if you give it to the person who's going to respond, it may be a very good treatment. There's a factor 12 monoclonal that's in clinical trials. It's finishing phase 2, will be going into phase 3. That looks very promising. There are companies that are looking at knockdown strategies, either antisense or interfering RNA. It could be, everybody so far is concentrated on plasma kallikrein, but there are some companies that are sniffing around doing this for factor 12 as well. Gene therapy is being looked at. I have some qualms about that, but I don't have time to talk about it. And then gene editing, I think down the road is going to be an interesting strategy, CRISPR-Cas or whatever the gene editing technique du jour turns out to be by the time it gets to HAE. And finally, the area that I talked about that we're interested in, C1 inhibitor rescue, has some potential. I think it's also important to say that there's an orally available bradykinin B2 receptor antagonist that's in clinical trials that is more potent than icatibant and longer lasting. And the company is talking about both on-demand and prophylactic uses for this orally available bradykinin receptor antagonist. And because I'm out of time, I had a couple more slides really talking about the pharmacoeconomics, but I'd like to stop there. And I don't know if we have time for me to take any questions, or we just end if it's a Zoom meeting.
This is Len. We'll certainly take questions. People who have to leave, but that was an amazing talk. A couple of simple questions. Do you hear me?
I can hear you. Yes.
When we say it's 1 in 50,000, does that include all the non-C1 inhibitor patients?
It does not.
I see about as many of those as I see type 1 and type 2.
Right, and I take your point, and the reason we don't include it is in the absence of solid diagnostic criteria, my feeling is nobody has a good feeling for how common or rare HAE normal is. In Europe, they think it's very, very rare. In the United States, we're deluged with patients who say, well, I think I have type 3 HAE. And the problem is how many of them are real and how many of them are not. Our feeling at the Angio-Edema Center as we're doing our test is we're, not everybody by any means, but a significant fraction of people we see, we think really have a non-histaminergic angioedema. So I don't know what that figure is going to be, and I think it remains to be seen.
Thank you. Another comment, this is just a comment here. I noticed David Hagan is on the phone. I wanted to welcome David and let you know you have an international audience. He's one of our former fellows and now faculty in Tel Aviv.
Okay.
David, thanks. The other question I had, after this session, I work with our fellows on a study group, and one of the textbooks we're using still talks about C2-kinin as one of the mediators of this disease, which I think has been debunked. Is that true?
I would agree that it's certainly not a significant mediator. C2-kinin is a fragment from the C2 complement component that's generated from plasmin and C2, so C1 RS. And you can show that it's formed in vitro. It's never been demonstrated to actually be made, that you have enough of those proteases in vivo. So nobody's ever showed that it actually exists. And we have very clear evidence that it cannot be the mediator of angioedema. So my answer is, it exists in vitro. You can make it. You can torture the system and make it, but it's not a mediator of an HAE.
Question?
Yes.
The ecallantide, Kalbitor, a lot of us think is more effective than Firazyr acutely. And one author I talked to speculated that it was because of the beta-1 receptor of the bradykinin, and I thought that was a little bit of speculation. I was wondering your opinion.
Yeah, I mean, it's certainly a possibility because a kallikrein will prevent the generation of bradykinin, where a contabon only prevents the binding of bradykinin to its cognate receptor. So I would agree that it's a theoretical possibility. The problem is it's a highly controversial area, and there's no good evidence that suggests that is actually what's happening in fact. And most of the development has been towards improving, as I mentioned, the bradykinin receptor B2 antagonist. There was an experiment done many years ago where a company made a combined B1, B2 antagonist. And the problem was the animals that got that actually had an increased amount of deaths from sepsis. They studied it in an infection model. So I think people are going to shy away from using both together. It's going to be tough to test that hypothesis.
Some people have no sense of humor about that.
Well, I think if we had HAE, we might not find the experiment very amenable.
Thank you.
I had a question.
Sure.
Yeah, what's for patients who we think may have type 3 HAE, what's the best place, again, to send them for more, again, either workup or what's the suggestions to do with the suspicions? Thank you.
Yeah, it's a really tough question. And so, I mean, you're on the West Coast. We're happy to see these patients. One of the things we think we're offering them right now is the ability to get both the TSKA assay we developed, but we're also routinely sequencing these patients. So if we get lucky, maybe we find the mutation, or the TSKA helps us arrive at a diagnosis, although it's a research test, so you can't really use it clinically. If our next study works, we would love to see our assay or somebody else's assay. We really need a biomarker assay that's out there that people can use to diagnose, because right now clinicians are in a total bind, and it's hard to get authorization to use, let's say, a drug like a contabon, which we like to use. We think it's a pretty good discriminatory way of saying, okay, you really do have bradykinin-mediated angioedema, but you really have to fight the insurance company to do that. You can look for the mutations. Invitae is the lab we use in the San Francisco area, but the problem is, it's pretty pricey. You can to ask for a custom group of genes, or to get whole exome sequencing and say, I want to know about these 4 or 5 genes. So there's no good answer other than to say, if you want, we're happy to see the patients, and we're trying to work towards, but so are other people, towards getting a biomarker out there.
It's just a follow-up question. These days with telehealth visit, are you able to do, the telehealth visit and then be able to draw the blood and then get processed? Is that something you've looked at? So, thank you.
So one of the things we've been studying is when we draw blood, because we're used to studying the contact system, everything is done immediately, temperature controlled, no plastic. We have very strict criteria for how we process the blood, and it gets frozen at -70 very quickly. We've wondered, will this translate? Can people send us blood? So we have been looking at how to perturb and how far off of how we collect blood we can go. And we think we've defined reasonably good parameters that would make it feasible for people to ship us samples. And we got that data shortly before the COVID shutdown. And so my lab's been shut down ever since, mid-March. And so we haven't started asking people to send us samples and see how it works. But I'm actually hopeful that we'll get to the point if people just do some simple, precautions and, treat the samples carefully, that we will be able to do things like telehealth and then mail down a plasma sample. It's no problem for the mutations. I mean, that's just DNA. It's pretty robust.
Any more questions before we let Bruce go? I had one. I noticed in your therapeutic options, whether we know which mutation or what the etiology of C1 esterase normal is, if you can get lanadelumab or icatibant approved, it's in my experience, it works. But you didn't list that as a thought process of how you would treat it.
So we do the same thing. If we can get it, we get it for these patients. We try to get icatibant for all of the patients that we think really do have HAE normal. The lanadelumab, I've seen mixed results on, and I actually would have more to say, but I'm constrained. So I won't say it, but I think I will make a prediction that not all forms of HAE normal will respond to lanadelumab.
All right, well, that'll be next year's talk.
And I'm sorry that I can't say more right now.
That's perfect. Thank you. Any more questions before we let Bruce sign off?
Thank you.
Thank you.
Well, thank you. I have to say that it's hard. I don't know how many of you guys have given talks by Zoom. I found it very difficult to pace myself and to, it's nice to be able to see an audience. I wish I could have been up there. I've always enjoyed my trips to Seattle, and I hope in the future that I'll have an opportunity to come up there again, because I think you have a wonderful group, and it's always a pleasure meeting and talking with you.
Those days will come again, I certainly hope. So thank you again. Everybody will be back together next week with another speaker. Thanks, Bruce.
Okay, take care.
Bye-bye.
Stay well.